Thank you very much for joining in today, ladies and gentlemen. Today we have Professor N.K. Udaya.
Yeah,
N.K. We have Professor N.K. Udaya Prakash ji. He's from Chennai. Some years back I went to his lab to study about airborne fungi, aerobiology, bioaerosol. So it was a very good training. He has kindly agreed to give this talk on airborne fungi. Before he actually starts his talk, I request Romana to give a brief introduction of Dr. Udaya Prakash ji to today's audience. Yeah.
Good morning, everyone. It's a great pleasure for me to introduce such a dynamic personality, Professor N.K. Udaya Prakash sir. Today, he will enlighten us on very interesting topic, aerobiology. Dr. Prakash did his PhD in aeromycology from University of Madras and postdoctoral fellowship in aerobiology and microbiology from Asthma and Allergy Research Institute, Chennai, and R&D Kevin Care, Chennai, respectively. He has 19 years of teaching experience and industrial experience from India as well as abroad. To his credit, he has various innovations in the field of aerobiology, microbial biotechnology, herbal biotechnology, bioremediation, bionanotechnology, etc. He is a member to various accredited bodies and is awarded to 11 fellowships. He has published 95 research articles and has organized 20 workshops and training programs. With this brief introduction, I welcome Professor N.K. Udaya Prakash sir to take over the platform. Please, sir.
Thank you. Thank you for your introduction. Let me know whether I'm audible to you.
Yes, sir, you are audible.
Yeah. Yeah. So before I start, my heartfelt thanks to Chennai and its team members, specifically the MycoAsia. They're working 24/7. Even I get plenty of disturbing messages, whether either it is day or night. Kudos to the entire team. They're working hard on propagating the field of fungi. Of course, even plenty of you are contributing your time on Sunday to learn about the airborne fungi. That's great to see. Before I start, let me say how many aerobiologists are there in India. Full-time aerobiologists, when you're thinking about, even about the society, you can see only around 200 odd members for the entire country. So this is what the demand of the aerobiologists is. Again, it is not much probe field, but there is a plenty of opportunity for you, the youngsters, to learn about. Why I'm speaking in that angle is just this is for beginners. So I'm making that set. Right. This is about the introductory slide, which speaks about my affiliation towards Wales University and with Marina. Before venturing to the airborne fungi, let us know what bioaerosols are. Any biological material suspended in the atmosphere in the form of aerosols, then they are termed as bioaerosols. It includes bacteria, of course, our own fungal spores, actinomycetes, fern, pollen, algae, plant cells. Now it is being increased to the proteins as they are the biomolecules, enzymes. Where we can see these protein and enzymes, of course, in plenty of biotechnological industries, and in processing, it is becoming airborne. Similarly, the endotoxins and mycotoxins, apart from that, volatile organic compounds, which is again emanating from the fungi. Now, I'm also working on with the airborne nanoparticles, which are of biological order. Although they doesn't fall under the category airborne fungi, just it's an introduction about what the bioaerosols are. Towards aerobiology, its history, it dates back to biblical literature and in our own Vedas. Or they say the disease of plants, even you can see that is spreading through the grape and then vineyards. So these are all the things already seen in biblical literature. Coming towards the history of aerobiology in India, it is by 1883, the Britisher Cunningham. He has studied the airborne microbes in jails of Calcutta. Why it is? It seems that the fever is spreading from one cell to the other cell in this prison. So he got a doubt why the disease is spreading across the cells. So he raised, oh, this may be due to the airborne nature of a specific disease-causing organism. So that is what the written literature we came to know about by Cunningham by the year 1883. Towards the history of aerobiology, it is Pierre Michael who is the first one to study about the airborne bacteria and fungal spores by the year 1850. It is Pierre Michael. So the age of aerobiology is concerned, it is around 175 years, according to the papers. Then by 1873, again, Cunningham from India, where he used aeroconicoscope to view about the airborne microbes. The aerobiology is the term given by Fritz C. Mayer by the year 1938. Later, there's a break in the research towards aerobiology. However, by 1973, starting from that till 1992, P.H. Gregory, he has did plenty of work on aerobiology. So rightly, he's called as the father of aerobiology. It is P.H. Gregory. I'm a receiver of P.H. Gregory Award. Of course, my professor, B.P. Vittal, is trained by Gregory. So I feel pride in as that I'm also trained by a student of Gregory. Later, plenty of work again is carried by Lacy in UK, where he did plenty of work related to indoor and outdoor air. In India, it is Professor S.T. Tilak, hailing from Pune, who did plenty of work. And he has produced more than around 50 students in aerobiology alone. So rightly, he is called as father of Indian aerobiology. It's Professor S.T. Tilak. Coming to as the classification of the atmosphere, where we can work. Of course, if you have the capacity to reach the exosphere, which is about 10,000 kilometers from the Earth's surface, that's called as exosphere. Then the thermosphere, where you can see the shuttle and the aurora, which is around 100 kilometers away from Earth's surface. Then the meteors, that is called as mesosphere, 85 kilometers. Then stratosphere, our weather balloon, when we want to study about weather. So they are reaching up to 50 kilometer in the sense it's stratosphere. However, most of our work are confined to troposphere, where even our flight can reach. So this is the troposphere, where plenty of activities are taking place towards the airborne fungi too. So this is what a stratification of the atmosphere. This is classified into troposphere, stratosphere, mesosphere, thermosphere, and exosphere. Branches of aerobiology, is it confined only to the fungi? No. Broadly, we can classify the branches of aerobiology into intramural and extramural. Intramural in the sense when you're working with the environment which is enclosed with walls on all sides. Whereas extramural, there is no such boundary. Again, these intramural and extramural can again be classified into different areas depending upon the area where you work. When you work with the pollen grains, it's called as the aeropollenology. Of course, Marina Labs is again having a specialty on this area. Aeromycology, of course, our pet area. Aerobacteriology, yes, we are working. Aerovirology, we haven't ventured into this so far. However, my student has reached University of Florida, and she's used to work on virology, and she's the first one to isolate the COVID-19 virus from University of Florida in US. These are all the examples where you can see intramural and extramural environment. Home, yes, again, it is confined by walls on all sides. Hospitals, hotel, any industry, place of worship, educational institutes, animal houses. We are taking it into the transport system even, because the bus, train, everything. Now it is Vande Bharat. Here to do the research on it. Malls, theaters, everything. Towards extramural is concerned, the garbage dumping site, where you can see there is no closure, or it is not enclosed into any borders. Markets, highways, agricultural field, even the entire city representing Chennai or Mumbai or any cities, and public environment like parks, This falls under extramural. This is all just an example. Of course, whenever you are finding about with your environment, just classify them under either it is extramural or intramural. It's a beautiful science where we call it as interdisciplinary science. Of course, all the academicians are advised now to work with interdisciplinary science, and plenty of saying a career is associated with this. We are calling the aerobiology as a typical interdisciplinary science because it's having an impact on human diseases, agriculture, forestry, finding a role in pollution, even the meteorology, defense organizations, forensic science, outer space, animal disease, biodeterioration, even the movement of locust, palynology, and also it speaks about general ecology and phytogeography. Again, it is not limited to the list what I have mentioned here. So it's a beautiful interdisciplinary science. And of course, I feel pride to say that I'm an aerobiologist. The pathways, of course, this can be, the principle of this aerobiology can be classified into 5 major stuff. 1, source. Whenever you are finding about any airborne biologicals, the source is surely next to that. If there is no source, they will not come into the atmosphere. They release, yes, they have to be released to the atmosphere. So they need a wind or any external disturbance so that they can have the flight into the atmosphere, and then they deposit whenever the weather factors are quite good for them. Based on the deposition surface, they have an impact. For an example, if they are going to reach your alveolar system, they can cause allergy or some other disease. If they are going to face a deposit on the leaf surface, if they are pathological enough to cause a disease, they may form a plant diseases. Each and every stage of this aerobiological pathway is controlled by the external environment factors, which includes the temperature, humidity, rainfall, the air current, wind speed, everything plays a role over there. This is just an example where you are seeing that the mold is growing on the doors of molecular biology laboratory. What we feel, oh no, every laboratory needs to be maintained with biosafety level, this, that, everything. But you can see that the entire door of the molecular biology laboratory is colonized by the fungal species Clarospore. And again, other indoor environment where you can see the fungi are growing plenty. So just think when your kids are playing hide and seek in this area, what will happen to them? Exposure to these fungi. Apart from that, they are well colonized in this area because of the moisture and they are thriving good. So you can expect volatile organic compounds, that is fungi-based VOCs also in this environment. So we just talk. Is a bathroom. Whenever you're bathing, just you're spilling the water onto the top of the roof and jumping onto that to have some excitement. So if you do that, you can see again the growth of molds on surfaces. Let us have a closer look. This is how they look like. They're not even living the household items. This is an apple. Somebody say it as an apple. Whenever I'm exposing this to students, first they think it's an apple, but it's a tomato colonized by Alternaria. In the onion by Botrytis. Chili again by Alternaria. And the lemon beautifully colonized by penicillin. A disposal. So you may be knowing about the source, where they are. So they can exist anywhere, provided the moisture, the food, everything is in favorable condition. Dispersal is concerned, wind, rain, insect, animal and human activities, they can easily disturb this. The image 3, 4, and 5 speaks about the airborne pollen grains. But the first 2 images speaks about the airborne fungi. Yeah, dispersal. And then their flight. the flight of these spores depends upon the size, shape, and their bright nature. This is beautifully depicted, diagrammatic representation, where you can see tetraploa on the extreme end, then Curvularia, Helminthosporium, Drexlera, alternaria on the middle, pair Trichoconus and Trichoconus cluster on the extreme end, and then the smaller sized ones as the penetrator. Again, they are classified into xerospores and gliospores, depending upon their nature towards dryness. So all these factors, the moisture, the size of the spore, even the shape, I can say. For an example, if you are taking the fusarium, it's a sickle-shaped spore where it is not easy for them to be carried to the atmosphere. So mostly you can see them as a soil-borne fungi rather than the airborne. But we are recording fusarium also into the air. So there is a controversy going on behind. But the spores of fusarium are sickle shape. How come when the wind is hitting onto that, it may fall down based on the shape of it? But the beauty of fusarium is they produce two types of spores, namely the microspore as well as macrospore. When we are speaking about the sickle-shaped spores, it is speaking only about the macrospores, whereas microspores are small and they are not having the shape of that. They are even ellipsoid. They can easily carry it to the atmosphere. So we are recording fusarium in the air too. Its impact when they are becoming airborne, what they do? Right, let us travel towards this. Beautiful colonies of Cladosporium. I like this particular picture because the closure view, which was presented to me by André Labbé from France. Then volatile organic compounds. So don't think that only the spores of the fungi alone can are becoming airborne and they can cause the impact. The volatile organic compounds, whenever they are thriving, the secondary metabolites, they are coming onto the atmosphere as volatiles. Even they can have the penetrating ability onto our system and they cause wide range of diseases. I want to highlight this particular fungal species, Aspergillus fumigatus. The beauty of this fungus is it's a thermotolerant one where it can grow from the temperature of 20 degrees, then it can reach up to 55 degrees. So if you're exposing this fungus to 20 degrees, yes, they can colonize, or to 55 degrees, yes, they will colonize. So when we are talking about the optimum temperature, it can easily fall by 37.5. That is the human body temperature of nearly 38 odd. So again, the next advantage of this fungus is they're having the fungal spore size of less than 1 micrometer. So they ranges from 0.8 micrometer to around 1.5 to 2 micrometer. So they can easily reach our alveolar zone through our respiratory system. So when the fungi are becoming airborne, what will happen? Actually, we can speak a lot about the pathogenesis caused by the fungus, everything, but they're all not by airborne. As the title is restricted to airborne fungi, I'm confining myself to the airborne diseases caused by these. So when they are becoming airborne, the route of entry is only through our respiratory system, not through contact. Of course, contact, but this is negligible. Whereas you can control dealing with soil, you can control dealing with water, but it is not easy for you to control whenever you are exposed to the air. So without any resistance, you may be inhaling the airborne fungal spores. Here I want to call your attention about the nature of or the color of the fungal species. Aspergillus niger, black in color. Aspergillus flavus, green in color. Trichoderma, green in color. Then again, fungi can be classified into hyalinaceous fungi and then demicaceous one. In the sense, demicaceous one have plenty of pigment, specifically the melanin. That clearly shows that each and every fungal species is possessing different protein composition. So when different protein composition are getting into your system, your body is challenged so that they can easily induce allergy. Again, the route of entry is through our respiratory system. So we'll be having the allergy related to respiratory allergy. So they can easily induce us asthma, chronic cough, sinusitis, bronchitis, pharyngitis, even the running nose, and COPD if they are well grown onto your lungs. ABPA, this is particular stuff that is obstructive bronchopulmonary aspergillosis. As I previously said about the Aspergillus fumigatus, they can easily reach your alveolar zone as the temperature is conducive and even the moisture provided, they can start colonizing your alveolar zone. So if they are continuously colonizing, then that can result in ABPA. Again, aspergillosis and aspergilloma are the disease nomenclature given for this particular stuff. And again, don't confine what this is caused only by Aspergillus fumigatus. If you are immunologically weak or otherwise, these fungi as a secondary infection, for an example, even in COVID, so the fungi, you might have heard about even mucormycosis. That is again airborne. Or a lot of controversy again in that mucormycosis. That is, there are plenty of people are speaking that it is because of mask. No, I can easily say as an aerobiologist, and again, I'm watching the disease continuously. I can easily say that it is not through mask, but only through the cylinders what we have used. Because the mask, even in COVID wave 1, plenty of people used. The same thing happened in COVID wave 2. So there is no difference existing. That is the cause of the mucormycosis in wave 1. Only by wave 2 we got. By wave 2, there is a plenty of demand for the oxygen cylinders. What we used, we have gathered the cylinder wherever it is, and we are not checking about or purging about the quality of something. Just we used to pump in the oxygen and then to give it for the patient. So the leftover cylinders, they are rich in oxygen, and they are also found to possess that is moisture. So mostly the Raju and the other mucormycosis, mucoraceous fungi, they will be growing onto the cylinder without purging them or quality check. Just we are pumping in the oxygen and giving it for them. That is, in the sense, we are inoculating the fungi for the patient who are already compromised or infected with COVID. So they cause secondary infection. That is what the mucormycosis resulted in. Again, this is restricted at the nasal zone because of the size of the fungal spore, where they are producing the colony on this nasal zone. Then mostly it is affecting the eye and then the ear. Right. Here we are going on to the volatile organic compounds. Just forget about it. If the fungus are smaller enough or less than 8 micrometer or something, for an example, I can take it as an actinomycetes. So they can easily reach or break the blood-brain barrier. When they are doing it, they can easily reach onto your blood system, circulatory system. Here I want to recall one of the central minister who underwent the open heart surgery. He died after a couple of years because of the fungal infection in his bloodstream. Then how come the bloodstream is possessing the fungi? That is because of the spores. The spore size, they can easily reach into the system then when they are going. Even in my laboratory, from where Dr. Sashireka may be knowing about Professor Indra Kalyanasundaram's lab, where we used to work plenty on Aspergillus flavus, and few people have reported Aspergilloma. In the sense, the presence of fungal spores in their bloodstream. Right. Again, these volatile organic compounds, when you're continuously inhaling, they can easily reach your brain. Just think about the smoke or something like what we're doing. They can easily reach your brain. Then they can easily attack your nervous system. So it can easily induce the Parkinson's disease. That is because of there is a disturbance in our neural system. They can also induce Alzheimer's. And again, the symptoms I don't want to say about, because most of us may be knowing what is the symptom between the Alzheimer and then the previous, that is Parkinson's. Even they can reach the renal system. So the spores or the fungi which may grow onto your kidney can cause renal malfunction. They can easily block the capillaries. So the neuron, or sorry, here the nephrons, cannot work or they cannot filter, so resulting in renal malfunction. So this is all because of the airborne fungi. Again, when you are genetically disturbed, even it may cause mutation. So mutation results in symptoms of down. Coming towards the air samples, how you can monitor these airborne fungi. There are plenty of samples available. And of course, I'm feeling proud to say that Marina Labs is the richest one in India. No other institution will have this much of wide, diverse samples to monitor the airborne fungi. The first simple method is sedimentation method. That is just you open the glass slide or the petri dish with the media, leave it in an environment for 15 minutes or something. Then pick the petri dish, incubate it. Later, by 3 days or 4 days time, you can see plenty of fungal colonies growing onto your petri dishes. But here we cannot give the quantity-based survey, whereas the quality can be easily said. What are all the species present? That can be done using gravity petri dish. Gravity slide is concerned, just take your glass slide, apply some Vaseline onto that, leave it in an environment for 15 minutes or maybe for 24 hours even. Then take the slide directly to the microscope, stain it with lactophenol cotton blue, then view it. You can see plenty of spores deposited on the slide. Again, the diversity can be known. Here, what's the difference between the petri dish and the glass slide is, it is a known fact that not 100% of the spores are viable. Not all airborne fungal spores are viable. Whereas when you're using gravity slide, whatever the spore deposited, even the non-viable spores can also get deposited, which can give a clear count on the presence of fungal spores in any environment. So this is what the difference. So gravity slide, that gives you the count of either they are viable or non-viable. Whereas petri dish exposure, they give only the fungi which can be able to grow in the media what we have given, or they need to be viable in nature. So these two are the major factors. Right, inertial methods. Let me go on to the next one. That is inertial in the sense, just keep the trap as such, allow the air current to come and deposit on the surface. Just keep them straight or whichever the way you want. Allow the air current to come here and there. Mostly in outdoors, we are using a buoyant sampler for indoor, and again, even it is considered as a personal monitoring one. Vertical cylinder trap to trap any external environment or extramural environment. So it is classified into airflow impactors in the sense we'll be using an external device to suck the air onto our sample. So Burkhardt personal sampler, maybe by next slide you can see the images of that. Burkhardt sampler, aerosol cassette, liquid impeller, and Anderson samplers falls under this category. Segmentation methods. These are all the examples. Just open petri dish. Then again, a glass slide which is coated with Vaseline, just to remove the peel the sticky surface stuff, expose them, can get the deposit. And inertial sampler, what I said is vertical cylinder trap. Just take the glass cylinder, wind up with the cellophane tape, again coat it with an adhesive, leave it in such condition for 24 hours, then peel the tape again, take it to the microscope, view what all the fungal spores deposit. This can be done throughout the year, every 365 days. Every day you need to change the cylinder or the adhesive tape. And here is the airflow samplers. That is the sampler falling under the third category. Liquid impeller. This costs you around 13,500 in India, whereas these are all things imported from US. So it cost me around 28 to 32,000 because they are NABL certified volume-based sampler what I have. Then the second image you are seeing is Anderson sampler. Again, the problem in India is as there is no takers, there is no manufacturers. Even if you are manufacturing, only we have around 200 full-time aerobiologists who are working on this. So selling capacity is very low. So marketing this stuff is more or less completely challenging. So awareness on this subject is not created. Then the RCS, which is made in Germany, the others, it's functioning with the other system. Then the biosis, here we can use just a glass slide coated with another aerosol of the same principle. Cyclics, again, work a personal sampler. Everything is all four falls under the same principle where you can expose just a slide coated with another. Take it to the microscope directly. See. Other advantage of this is you can give the result by 20 minutes time. Whereas in culturable ones, it will take a minimum of 48 hours. Whereas the client cannot wait. So if you want to monitor any industry production unit where they are expecting the result by 20 minutes or 30 minutes time, the images what I have given on the second line are suitable for it. Let us travel towards outdoor. Traffic junctions, the highways, this falls under outdoor. Again, our sample at work in one of the traffic junctions in Chennai, and again in Marina. So of course, for us to go about and to get relaxed, this is in Mamallapuram. Of course, it is a UNESCO certified site. We worked a lot on these coastal zones. Sample at work. Occupational sites is also here. We have just saw about the people exposure, and over here occupational environment. The farmhouse is a beautiful one where you can see plenty of organic sources available for the fungi to proliferate, and they can easily become airborne. More than 3 tons of cattle dung deposited on a single spot, which is conducive environment for the microbes or the fungi to grow. Whenever they are disturbing it, plenty of thing comes onto that. In this area, when you're comparing with that of fungi and actinomyces are very high in numbers in these areas. Our sample at work. In the other side, vegetable market, where the waste vegetables are thrown here and there, the gunny bags colonized by fungi, again, with possessing plenty of moisture. So they play a major role as a source. Then again, human disturbance. After so plenty of fungus come into the atmosphere. So the people working in this environment are rightly challenged with the airborne fungi. So samples at work. Pocket. Moisture is there. However, when we studied about the airborne fungi and bacteria in this environment, bacteria is too high when compared with the presence of fungal spores. That is because of this is not a continuous source available for the fungi to proliferate. Every day they are cleaning it. So not much fungal spores are recorded in this. I've used the impinger right here. So you can see how the impinger is used in monitoring the airborne fungi or any airborne bacteria or airborne microbe. So this is connected with a small sampling pump, which can suck the air at the rate of 5 to 10 liters per minute. In the impinger, I can use any supporting media, or otherwise even just a buffer or whatever the media I want. So I can expose it in a liquid form, take it to the laboratory. I'll do the serial dilution so that I can give the count on the presence of airborne microbe in this environment. It's a beautiful environment that is where I can see a cocktail of fungi. One of the most disturbed environments where more than 55,000 people are engaged directly or indirectly. That includes the rat pickers too, the lorry drivers, cleaners, garbage collectors. So plenty of people's risk, their health is at risk. Again, I have recorded Aspergillus fumigators as the second most dominant in this environment. Even my personal interview with the personnel working in this environment, they used to say they are mostly affected by TB, tuberculosis. The sense that they cannot be able to differentiate aspergillosis and then the tuberculosis. So either way, we need to give a lot of concentration to these people, and then the government need to support on these projects to know about the health of those people, because they are working for us. In our samples at work in this environment, you can see that the lorry drivers and plenty of lorries are carrying the garbage of the city. Chennai alone arises around 4,000 tons of garbage in a day. Let us get into the intramural environment in the sense of the work environment, which is confined to the walls. So you can see just a kitchen or a bakery where the person is working. There you can see the dough, that is again the cellulose-based material. Here we are recording Aspergillus niger as a dominant one. The guinea pig house, again a beautiful environment which possesses a mixture of sources. The guinea pig are fed with the cooked pulses, then the straw as a bed. It is wetted by the urinals of the animals, and then the dung pellets. All these things serve as a source for the fungi to proliferate in this environment. Apart from that, they are supposed to be maintained at 28 degrees continuously 24/7. So moisture is also there. The conducive atmosphere is provided for the fungi to proliferate in these environments. Similar is the condition with the rabbit house. Also, the animals look cute. Whenever you are entering into this environment, you may feel uncomfortable because of the urine smell generated. That is, plenty of ammonia are generated along with the presence of fungi. So the people or the animal caretakers who are working in this environment are continuously challenged with the presence of airborne fungi and then the secondary chemicals, that is volatile organic compounds. I have not worked with this particular environment. If any of you attendees are willing to work in this, I welcome, because I'm scared to work on this rabbit house. Now, can I get an answer? Is that a rabbit house? Because I can sense plenty of people are feeling sleepy, so I want to pose a question. Is this rabbit house? Am I right? No answers. Oh, typing. Yeah, I'm waiting for your answers.
It's a lion's enclosure.
Is it lion or tiger?
It's a tiger, I guess. Yeah.
A tiger. That's great to know. So whenever I'm posing a question to my students, they used to say it's a tiger, but it's a lioness. So that shows how we are active. We need to take our kids to zoological park at least to differentiate the lion and tiger. Right. So this is what our kids are. Then where we can study about the fungi. They are clearly visible, but airborne fungi are invisible. So kindly give your attention for that. Right. So again, I want a person to work on this. A couple of my veterinary students, they did their research, that is their final year project, about the airborne fungi in the wild animals, den or house, I can call. Food preparing unit, Anderson sampler at work. This is the bakery, the huge one. You might have heard about the modern bread. So this is modern bread factory. So continuously 24/7, this is working, and we started studying about the presence of airborne fungi in these indoor occupational environment. This is the beautiful environment where the people are continuously challenged, immunocompromised. They are moved on to the ICU. If we are speaking about the ICU, then my attention goes towards the air conditioning system, how good they are. Because the filters, are they frequently changing, or they are the best colonizers? Because I worked a lot on air handling units in Singapore and in US, and they are most of the culprit. Even in Chennai, we worked with Nokia, where their employees are getting affected. So we started studying about the filters of AH. Again, plenty of microbes are growing in AH, where I can see the presence of Aspergillus fumigatus, Acrimonium. So these are all the small spored fungi. They are growing aplenty. So these fungi are again thrown continuously into the atmosphere in ICUs. So the people who are admitted onto that are again continuously exposed to the airborne spores. So this is one of the client in Karnataka. They gave us this particular project to monitor about their hospital. And then this you can see January, February, and the third one is this, where a couple of my students in Pondicherry, they did this research to monitor the presence of fungi in mortuaries. But no result we got, again, because plenty of disinfectants are used in this. Again, frequent cleaning. Because of that, we are not recording that much of fungal spores in these environments. Right. Fungi as a pollutant, again, it's more or less a recall. Three-dimensional attack. They serve as a pathogen, oxygen, as well as the allergen. Size, they falls under 1 micrometer to 50 micrometer. So they have an ability to penetrate your respiratory system. Either it may be upper respiratory tract or otherwise a lower respiratory tract. Source availability, it depends upon the presence of fungi, depends upon the availability of the source. They need to have the capacity to be buoyant in the atmosphere. Then their number need to be significant. Then we need to be careful with these fungi. And just a few things about the count, what I have recorded. This is again a vegetable market, what I previously showed you. Here you can see the Aspergillus niger contributing around 1,520 colony forming units per cubic meter of air. In the sense, so in 1,000 liters of air, 1,520 Aspergillus niger spores are there, viable spores. This is followed by Aspergillus flavus, and again the third dominant is Rhizopus, which is a Mycorrhizal 1. Totally, I've recorded 2,050 colony forming units in a cubic meter of air. That is, if a person is working in an environment for continuously for 2 hours and 40 minutes, he is inhaling 1,000 liters of air. So if a person works in an environment for 8 hours, he is nearly inhaling 3,000 liters of air. That is, the person exposure in a vegetable market when he is working for 8 hours is around 6,150 colony forming units of airborne fungi. In rabbit house, you can see Aspergillus flavus is dominating, followed by Aspergillus niger, whereas third dominant is again Absidia, which is again a Mycorrhizal fungus. Total average CFU is 705. In garbage dumping site, total average 733. Dominant Aspergillus niger, followed by flavus, whereas the third dominant is Aspergillus fumigatus. So every day, the person is exposed to around 300 viable spores of Aspergillus fumigatus when he is working in solid waste dumping site. Again, I want to recall that when you are checking with the WHO's critical care species or microbes, they have listed Aspergillus fumigatus as critical care pathogen 1. So high priority is given for this particular fungal species. And where they are free of pollutant, that is particularly the airborne fungi, our study shows that it is towards the coastal zone. We have studied nearly 33 coastal zones, starting from Yennur to Mamallapuram, covering around 60 kilometers. So every 2 kilometers, we started studying about the presence of airborne fungi. Finally, we have recorded only 20 colony forming units per cubic meter of air. So if you want to get or inhale a clear air, which are free of fungal spores, taking a troll at coastal zone is good. So don't miss the seashores. Visit Chennai. You're always welcome to Marina as well as Marina Labs. Right. So this is what a simple book I have written right now by the month of March. And again, it is read by 1,092 scientists throughout the world who are distributed in 22 countries. Again, it is available free of cost. For any amateur mycologist, this may be a handy book. Using a simple tape, they can monitor the indoor molds, which speaks about how you can take the sampling, how you can take it to the microscope, then how the images may look like. It is having a pictorial atlas. Again, it is free of cost. You can download it from my ResearchGate profile. We are also offering a diploma in aerobiology, which courses paper 1 and paper 2, and again practicals. Either it may be in the form of online or offline. If you are interested, you can mail to marinalabs@gmail.com. And if chance and time permits with the MycoAsia, I can come about with the next title as fungi in natural traps. That is, again, the natural traps are serving as a trap for the airborne fungi. Before I finish, thanks to Dr. Shanai being instrumental in bringing about the excellent forum MycoAsia, and fellow colleagues, plenty of scientists and other team members are working behind this, I know. And then all the listeners of this today's talk are showing interest on this and contributing your Sunday's time specifically to learn about the airborne fungi. Thank you again. Thank you, Dr. Shanai, for giving this opportunity. Any queries is welcome.
Yeah, thank you, Dr. Udaya Prakash ji. Let me unmute the people, actually. I think they are kind of, okay, now they can speak up. Any questions, please ask. There is one question in the chat box, Dr. Uday. Is there any free courses available on aerobiology?
Actually, we are offering the course as free course, but for certification alone, we are charging.
Okay. Thank you.
Good afternoon, Professor. This is Dr.
Yeah, Shyam.
Shyam Kumar Rajaram
Yes.
from Kamaraj College of Indian Technology. Thank you for such a wonderful program organized by Dr. Sanai and special lecture given by Dr. Prakash. And I have little clarification regarding the aerosol. Actually, during winter or even in summer, and when we open the refrigerator, at the door there is a gasket. In that mean space, lot of fungal growth. Is it cause any defects to the food material which we are keeping? Is it the infection or the contamination by aerosol or through food, doctor?
Not as aerosol. Actually, they fall under the category psychrophiles. So they are having an adaptive nature to grow at lower temperature, mostly at 8 degrees centigrade. So if you're carefully examining, you can see that the acrimonium species are having that ability to grow at that. Even you can conduct such survey on plenty of fridges which are present throughout Madurai. Yes, we can go about the diversity of them. So that may also be a good piece of research. So it's not a airborne, mostly because of the condensed stuff present within the refrigerator and contaminants. They are making these fungi to grow.
Okay. Thank you. Thank you very much.
Good afternoon,
Dr.
sir. Sir, good morning.
Yep.
Please raise your hand. I will let you speak. Thank you. First, I will request Mr. Archana to ask a question.
Sir, I am from Nagpur.
Yeah.
I am student of Dr. Mrs. Sauji. I have also pursued my PhD long before in aerobiology. Sir, I want to know, here in India, are there any standards so that we can say that this much CFUs per liter of air are safer side or they are harmful?
Yeah, it is. Around 500 CFU is permitted. But once I have raised the question, because we are just following whatever the prescription given by Western countries. For them, the temperature, everything, climate and weather are different. We cannot adopt the same for India. So we need to bring in, of course, our Indian Aerobiology Society need to work on this. I've raised this question long before, nearly more than 15 years before. But till date, we are not getting the clear-cut answer from Indian Aerobiology Society. So we need to work on that.
Yes, sir. I search a lot, but I cannot find the CFU standards for India. So it is very necessary for us to set the standards. Thank you so much, sir.
Thank you, Archana.
Mrs. Sweta Belosi, please.
Yeah, good afternoon, sir. Sir, I'm from Mumbai, Ratnam College, and we have undergraduate students, and we give a lot of projects based on fungi. So, if is there any key, like fungal spore key, through which at undergraduate level student can identify, and we inculcate research papers presentation at undergraduate level. So, is there any key available, sir, for fungal
Yeah, yeah,
spores?
yeah,
And
possible, ma'am. Even I have written a book on indoor moles by the year 2004. So that
okay.
book can also serve as a reference material.
Okay. So,
Then
so, so,
Professor Tilak has also written many
huh.
books on airborne fungi. They are possessing pictorial one, or otherwise with the diagrams based on the spore size and shape of it. You can easily identify them. If you have any problem with identification, just kindly send out the image. I'll try to identify with my
Thank you very much,
expertise.
sir.
Yeah.
Thank you so much, sir.
Yeah, sure.
Dr. Udaya Prakash ji, your video is not really streaming. Can you
Yeah,
try
maybe
to fix it? Otherwise, it's okay. No problem. No problem.
yeah.
Dr. Deva, would you like to ask your question? Yes, Dr. Deva. Dr. Devdatta, yes.
Yeah, first of all, congratulations for you joining the institute.
Thank you, sir. Thank you very much for your wonderful presentation. Sir, I would like to know, in hospital environments, we have yeast infections commonly. So in that cases, this airborne fungi can be useful. Like
In
how
hospitals?
we can trap them for yeast species, for yeast infections.
Yeah, I've shown plenty of samples, right? Even we have provided the consultancy for a few of the hospitals where they can monitor. Our advice is every 15 days they are supposed to monitor it, because most of the presence of the microbe in those environments are antibiotic resistance. Because as they use plenty of antimicrobials in hospital environment, whichever the microbe thriving in that environment, they have the capacity to have the resistance towards it. So they need to study by every 15 days interval. Apart from that, they are also supposed to do with the antibiogram test. So these are all the recommendations what I can make with the hospitals are concerned. There, when you drop to our lab, I can show you the samples, what we have and how you can do the sampling. So when you are visiting Chennai, just you drop in.
Okay, sir. Thank you very much,
Yeah.
sir. Sir, I would like to know any research gaps, because very few people are working on airborne fungi. So it will be useful for future researchers, like to explore on which areas we can.
Yeah, plenty of, particularly speaking, airborne virus, there is no work in India. It's a very wide gap. Again, with bacteria, because most of the people, again, with fungi is concerned, few of them are having a specialty only with the non-viable spores. Few of them are having specialty only with the viable ones. So these two has to be sorted out. Then again, third, a few of them are having a specialty on airborne pollen grains. So all three needs to be coming together. Apart from that, airborne bacteria, not much report or seen. Also, we can have plenty of microbiologists who used to work on the water and then soil, but not much on air. So again, there is a gap. What we need to explore is again with virology. That is what I say, because I felt very bad as we are not having a facility to monitor the airborne COVID. Also, I'm the first one to report the COVID virus as airborne. There are no takers. Even I have sent few of the papers stating about, to the journals, standard journals. What they say is there is no report so far available on the airborne nature of COVID, and they rejected my papers. What I said is conducting an yagna that can control the airborne virus. This is what the principle, what I said, because they are coming on to the air in the form of nanoparticles. They are easily clinging on to the COVID virus, making them to fall back. Right? Apart from that, if they are aromatic nature, they can even peep into the protein of the COVID virus. Again, they can make them ineffective. Or otherwise, they can peep into the COVID, disturbing the nuclear nature of the virus, so they can be killed. These are all the principles I have given, and I submitted to the few of the top journals, but they refused to publish it because they said there is no report stating that COVID has airborne. Only after 1.5 years or 1 year and 3 months later, they accepted, even CDC and WHO, they said, yes, it is airborne. So this is what the pity of science. And again, there are no takers whenever we are doing a real science.
Thank you, sir, for your
Yeah.
insights into this subject. Thank you.
Dr. Shafaq Khan, please.
Yes, sir. Yes, sir. Sir, I would like to collect recent information on aerology, sir. How
Yeah,
can I-
yeah, sure. Yeah, sure. Huh?
Yes, sir. Sir, I would like to collect the recent information on aerology, sir. How can
Okay.
I get this recent information, sir? In the form of books, papers.
Oh, okay. Just any site with stating the keywords aerobiology will do. If I have my personal collections, I can just share it with you. Just send your email where I can get it from Shenoy. Later, I'll send my papers towards aerobiology.
Yes, sir. Yes.
A lot of literature surveys we see nowadays, because when we did our doctorate, we had a problem because personally we are supposed to collect the literature from every individual scientist. Nowadays it is not like that. Plenty of open sources are there. It is easy for you to collect.
Yes, sir. Sir, because there is no books on this aerobiology in market, sir.
No. Professor S.T. Tilak has written plenty of books, nearly 3 to 4 books on it.
Yes, sir.
Mine it is 2. Again, one you can download from ResearchGate. The previous one that was again sold in 22 countries, but the charge is too heavy. 2,600 per book, so it is not available easily in the market. That is on indoor moles, isolation and identification.
Yes, sir. Yes, sir. Okay. Thank you, sir.
Yes.
Thank you. Okay. Thank you, sir.
Dr. Surabhi Sharma, would you like to ask your question?
Good morning, sir.
Yeah.
First of all, thank you, sir, for an informative session. Sir, actually, I want to do aeromycology certificated course. It is separated or combined with the aerobiology?
It's combined with the aerobiology.
Okay, sir. Okay. What are the charges, sir?
No, it's free of cost. Only for certification we used to charge.
Okay, sir. Kindly share the details on my
Sure.
mail. I
Sure.
will return the mail in the chat box.
Sure.
Okay. Thank you. Thank you so much.
First, ladies and gentlemen, Dr. Uday is in our WhatsApp group, in the MycoAsia network WhatsApp group. So we will identify in the group. If people can connect with him. Thank you. Yeah. Miss Neha Anton. Manohar, you can ask a question.
Hello, sir. The session was really informative. So I would like to know what are the preventive measures, to avoid these mold formations. And I'm much more interested on device because we are monitoring bacterial spores. So we are targeting bacillus species in the veterinary institutes.
Right.
So we are actually trying to detect bacillus species. And one more question is, what is the most commonly found bacterial contamination in the air?
I'll first answer for the second one.
Okay.
It's mostly the species of Micrococcus. I am recording around 7 to 8 different species of Micrococcus as a dominant one. Then towards limits of concern, no. So far again, there is no such thing brought in. To containment is concerned eradication of source. That is the major one. Again, it is easy in indoor rather than extramural environment. In your case, when you are working with the animals or the farmhouse, it is not easy. We need to just remove the sources whenever we are finding that there is a mold infection or bacterial colonization. Bacteria, if you are thinking about, then stagnant water or stagnant source with plenty of moisture, they are the reason for the growth of bacteria. So frequent washing of floors in those environment that can control the airborne. Yeah, Manohar, any other question?
Thank you, sir. So what about in case of fumigation? We usually do perform fumigations in the lab.
Right.
So sometimes still we may get contaminated, plates. So probably that could be from source, right, sir?
Right. Is that plate is taken for sampling or something?
Sometimes, yes.
Yeah. So that may be the reason. So seal it properly, and then you can take it so that it can be controlled.
Okay, sir. Thank you.
Dr. Shashiranga, come back, please.
Yeah. First of all, I would like to thank Dr. Uday for the wonderful lecture, and it's very interesting about this diploma course also, which he has said, because actually for aerobiology and aeromycology, I think probably many people are a little wary about this airborne things because it's not easily identifiable, if I am not wrong, and probably they are not having an access to sufficient literature,
Right.
and that could also be the reason. And then many a times there is a lot of other subject overload where this uncertain subjects are not easily digested or taken in, that. So we need to actually get some kind of a training done, or some small workshop, as you are doing it in your lab, I believe. You are doing these on not online. Online is very recent, but earlier also you used to do these workshops, right?
Yeah.
Training workshops where the children and students are exposed to the methods, how they are doing it, and where they can easily conduct, and how they can identify at their level. Simple ways, keys can be used to do that. Maybe that will make the subject a little comfortable for the students to get interested and then work upon it further. That this is just a suggestion. I mean, maybe we have to work all hand in hand together to get this awareness spread. I wanted to ask whether this protein, this allergy related to the protein of the this fungal thing, fungal spores, is it being conducted, or is there any protocol that we can do it? Because I have not done it so far, so I'm just asking for that.
Yeah, easy, ma'am. Easy. You might have seen, because when I did doctorate, you are my colleague who is also doing your doctorate. You might have seen me working with Raju hospitals in collaboration of taking the protein, and then I used to take either me or Bona used to take it to Raju hospitals to do that screen test. Yeah, so extraction of protein. But when right now we need to go in with the ethics committee. Previously, it was not there.
Yeah.
So if you want to avoid that, just what we can do is radioimmunoassay. So just extract the protein.
Yeah, you said ethics committee. Why ethics committee for proteins?
To do with the skin prick test, ma'am.
A skin test, okay.
Yeah,
Skin
yeah,
test.
yeah.
But the air can be sample. I mean, I am talking about the ones which we can culture, and then
Yeah,
from
you're
that
right.
we can
You culture
do
it.
protein. Yeah.
You culture it in mass. You extract the protein using the buffer. Now it is having the crude protein, having X or Y. Just you can run through SDS to know about the kilodalton, what sort of protein it is.
Okay.
Or when you want to find out whether it is really allergic, just you conduct radioimmunoassay.
Radioimmunoassay.
Right. Just take the sera of the people or patient, give it on the well. In center, you cut the well and have the protein of this our particular fungal species. So allow them to react. If the arc is formed, you can easily say that this particular protein is allergic to the candidate.
Okay. Okay. Yeah. So that is, there might be some chemical tests which are easily conducted, or it's only this SDS thing we have to do?
Using this alone, you can easily bring in even a kit.
Yeah, there are lots of.
Again, problem is when I did this research, even Dr. Raju asked to start me the pharma to bring about the protein of this fungi. But of course, financial strength and time, everything is needed. Our marketing potency is very rare. So the people who are working on allergy are again smaller in number. So also we did this. Marketing is not up to the mark. The work rate related to this is conducted by the Bose Institute in Calcutta, Shantiniketan.
Achha.
So few of the aerobiologists over there, they are working with this sort of protein and allergy. Mostly with palynology, they worked a lot, and then with few fungi.
Yeah, because see, actually in the air, everything is mixed. The pollen
Yeah, you're
grains,
right, you're right.
the bacteria, the fungi, fungal spores and everything. So
Yeah.
it's only through culture, you have to separate the cultures, isolate the cultures and then get all these things
Perfect,
sorted
perfect.
out, right?
Yeah,
I
you're right.
wanted to ask just one last question. You mentioned about this coronavirus and the fungus, some connection you mentioned. Like, I didn't get it very clearly.
Yeah, I was speaking about the secondary infection.
And the mycosis,
You're right, you're right.
Yeah,
You're right.
mucormycosis, which they were calling. But that is because of the what to say, the less resistance of the people. Right? It's only in comorbid people these mucormycosis, the corona infected people, they were getting it, right?
You're right, you're right. That's why I called it a secondary infection.
Secondary.
Yeah.
No, you mentioned something where the viruses attach themselves to the protein, something you mentioned. So I couldn't get what exactly
No,
you
that
mentioned.
that was to combat the airborne virus.
Huh?
What I said is even conducting yagna or towards south, we used to do that sambrani smoke or something, a type of fumigation.
Yeah, I understand that.
Yeah.
I understand that.
In Western countries, they use call it a smudging. So frequent smudging or doing this sort of yagna.
Okay.
So they produce the airborne nanoparticles. Again, this particular, the virus is, in my opinion, it is a airborne bio nanoparticle. Because the size is
they could attach onto these biological particles.
Yeah, you are right. So that they grow in size. They grow in size. If they are growing in size, they cannot sustain in the atmosphere. They fall down.
Yeah, maybe we could try a standard method for checking out whether it's attaching. Anyway, it's very interesting. Thank you so much. I think it has actually stimulated me. Maybe I'll think on that. Thank you. Okay. Thank you.
Dr. Vinay, please.
Hello. I read somewhere that if you can limit the humidity of the rooms to less than 50%, then there will be no growth of fungi. Is it so, sir? Because whenever we talk of limiting the growth of fungi in hospital environment, we are only told that you just take care of your ACs and that will be done.
But the problem is this, we need at least 65% of moisture. Otherwise, even our lung may get dry. So maintenance of that is not so easy. They can claim that reducing the moisture level to less than 50 so that you can control the micro, but that is practically, it's not easy.
So what is your suggestion to why I ask this question is because, sir, I work in Bhubaneswar. We are very high humidity area, almost sometimes it approaches 90%, 87, 90%. And the moment the central AC is switched off, the walls get flooded with water, and then the very next day you have fungus growing on it.
A lot of condensation.
Yeah, subsequent to condensation, overnight, 2-3 days, you have fungus growing on it, and then there are people who are worried about allergic sensitization to fungi.
That is, again, we can call it as sick building syndrome or indoor air quality related stuff, building related illness. These are frequently reported, but we need to completely work on it and know the go. But I don't think reducing the moisture to 50 degrees, 50% is easily work out, because again, you are supposed to handle with AHUs. Have you checked about the quality of AHU, whether they are changing the filters frequently? Because when I worked with Singapore, that was a major problem I had.
It is not done regularly, sir. It's not done regularly.
That may be the reason. But we cannot confine a single reason. Plenty of things are playing a major role in that. Just find out initially every step of it, so that it may be easy.
Thank you, sir.
Thank you.
Mr. Deepak Bhatt, please. Mr. Deepak Bhatt. Okay, Manohar, you have any questions?
Yes, sir. Yes, sir. So we do have a glass slide. On that, we do have a tape adhered on that. So I'll put it for environmental monitoring. So if there is any spores adhered onto the tape, so how can I redispense these spores onto the plate? I would like to grow them. So is it possible to do that?
Yeah, possible, Manohar. Just cut the area,
Okay.
place it on the petri dish with the media.
Okay.
Allow them for infection.
So the tape, the gum will be more stronger, right, sir?
No. Don't worry. You are giving supporting media, so automatically they start growing.
Okay, I'll try, sir. Thank you.
Mr. Deepak Bhatt.
Thank you, Dr. Udaya, sir, for a wonderful talk. So we got a lot of information out of this talk. So I have two questions, sir. So first question is, like my institute, there is centralized AC, and in the morning when we used to go, there are a lot of black spores fallen on the table or equipments. So what are these different species?
Are you sure whether they are fungal spores? Every morning you are recording?
Yes, sir, I saw, sir. These might be fungus spore or something else. But these kind of some aerosol sort of things which used to fall daily on the surfaces. So we need to clean up.
So there may also be with the dirt or dust particles which are falling from AHUs, that is the air ducts.
Okay.
So just pick them off, view it under microscope.
Okay.
Take the photograph and send it to me. Otherwise, even you can view whether they are the fungal spores or not.
Or
But
else.
I suspect it may not be the fungal spore. Every day you are saying it's falling, right? Because of the disturbance of rat and rodents on the air duct, so they may fall onto the table or something.
Okay, so second question is, my wife has bronchial asthma for last 2 years. So whenever there is cloudy outside, so these things get swollen up. So is this these allergen linked with some temperature or humidity kind of thing in the environment?
Yeah. Plenty of factors are there in inducing asthma. Even exercise can induce asthma.
Okay.
So again, this climatic change. Then typical location of yours. For an example, if you are staying next to eucalyptus, in some season, they may produce plenty of pollen grain, where your wife may be allergic to these pollen, or pinus, even parthenium. So the people from Bangalore, they always report they are allergic to the pollen of parthenium. So we have recorded around 13 different pollen grains which are good in number, inducing allergy. So that may also be the reason.
Okay.
But
So
it is
apart
not
from
for a single reason. Plenty of
Okay, so you're saying forced inhalation or air direction or other part with specific kind of pollens increase the intensity of this allergic reactions.
Right. Maybe.
Okay, so many factors are there. Okay, thank you very much, sir. Thank you very much, sir.
Sir, Udaya Prakash, please.
Sir, good afternoon. Am I audible?
Yeah.
Sir, thank you for that nice presentation. I just have a small query. You said that for these slides, we can take greased slides and we can put them. Then we can collect the aerospores from the air, and then we can put the lactophenol cotton blue and examine under microscope, right? So, so what about, sir, when we are taking a greased slide, how can we do the lactophenol cotton blue on it? Like it will it not be like will it be visible? Can we mount it?
Yeah, it is. Just have another slide on it.
Okay.
We have
we'll put another cover slip.
Cover slip, right?
Oh, we'll put lactophenol cotton blue and put a cover slip over it.
Perfect.
Okay. Thank you so much, sir.
Ladies and gentlemen, any more questions for Dr. Udaya? Okay, Dr. Udaya, can I ask one question to you actually?
Yes.
So I read in one paper that the fungal spores released from the Amazon forest, they help in the cloud formation, and they actually decide our climate. Is it kind of an exaggeration, or is there any evidence, like truth is there in that kind of research?
Want to be frank?
Yes, please.
Then it's an exaggeration.
Okay.
Because the presence of fungi is not that huge to induce the thing. The fact is that.
To
But
supplement,
they have
yeah.
a role, yeah, along with other pollen material, as in particulate matter, even burning of forest. So these things come onto the atmosphere. They start pulling the cloud because of their ionic nature. So surely it will be followed by rain. So this alone is the reason, but not as the fungal spores as a single individual.
Again, I think connected question. When the fungi have been made as a big villains in our home environment, especially in the dust particles. If you take probably 1 gram of dust particle, what would be the percentage of fungi in the bioaerosol component, like in terms of percent? Maybe just 50% or less than 50%.
Fantastic question, Shanai. Bona worked on this actually. So she did. She has collected the dust from 100 asthmatic residences. Then she examined the presence of mesophilic fungi as well as thermophilic fungi. There she recorded around 6,000 colony forming units per gram of dust. So dust is a very good source of the fungal protein. So whenever we are cleaning the houses, you can see surely there will be a person reporting dust alerts. So this is again not only towards the fungal protein alone. It is again a mixture of foreign particulates. So the protein is concerned. I haven't analyzed what sort of protein or how much the volume of protein may be in a dust. But this raises a question to work on this. What will be the percentage?
Okay, thank you. Just last question before I let you go.
Yeah.
See, like over the years, like hundreds of years, thousands of years, is there an increase in the number of fungal component in our atmosphere?
Seasonal wise, I need to check with the literature, but this is a voluminous one. Again, it makes me to work on this because the work what we did is previously by the year 1996 to 1998 in Chennai. So I can give you the data by that time about the Chennai, whereas if I am taking it right now, there will be a fall because of urbanization. So previously I had plenty of diversity of even plants. Plants are again a source of microbes. So even the leaf litters or something. As the litter fall is reduced due to urbanization, the fungi may not be that much diverse and even in count. So this is all due to the urbanization of any city. So whenever we are cutting about the forest or trees or plants, we are losing the diversity of fungi too. They're having a sequential chain of events.
I mean, this makes me believe that we need to actually continuously monitor the fungal spores in our environment, especially in outdoor environment where not especially, maybe we could start with metro cities and try to find out the relationship between the fungal component in our aerosol and with the industrialization or urbanization or pollution level. So I think it will be very much important to do this kind of exercise, I guess.
Right.
Yeah, thank you so much, Dr. Uday, and I request Dr. Romana actually to propose the vote of thanks. Romana, are you there?
Yes, sir, I'm here.
Thank you, Shana. It's a very great platform for me and had a very nice interaction with plenty of scientists and colleagues. Thanks for everyone.
Just wait for a minute. Dr. Romana will propose the vote of thanks. It's kind of an official thing. Thank you.
Good afternoon, everyone. Thank you, Dr. Udaya Prakash, sir, for sharing your vast knowledge on the very interesting topic, aerobiology. I would like to thank all the participants for being mutually active throughout the session. Thank you, MycoAsia and Shinoy, sir, for organizing this beautiful session. Thank you, sir. Thank you, one and all.
Yeah, thank you very much, ladies and gentlemen. Thank you, Dr. Uday. We'll see you around. Yeah, thank you.
Thank you, Shinoy. Bye-bye.
Good day, everyone. Yeah.